Hello guys, welcome to our channel Mushroom Your Knowledge. Today we are going to discuss about the structure and function of DNA polymerase 1. First question, why we call it DNA poly 1? The answer is because we know that now there are 5 types of DNA poly. It was the first discovered structure, so we call it DNA poly 1. At that time there was no evidence of other DNA polymerases. So molecular biologists assumed that polymerase 1 was only one that is responsible for DNA replication in prokaryotes. Now we know that the assumption was completely incorrect. Nevertheless, we begin our discussion of DNA poly with the structure and function of DNA polymerase 1 just because it discovered first and the structure is so simple. So first DNA poly 1 Arthur Convert discovered just Arthur Convert, note that name, discovered the first E. coli DNA polymerase in 1951. Although Pol1 have a versatile structure with a 102 kD polypeptide chain, it catalyzes three distinct reactions. The first one is DNA polymerase activity. The second one is 3' to 5' exonuclease activity, and the third one is 5' to 3' exonuclease activity. So the most important point is it shows the two types of exonuclease activity in the direction of 3' to 5' and in the opposite direction 5' to 3' exonuclease activity. Then the question arises, why does a DNA polymerase also need two exonuclease activities? The answer is the 3' to 5' activity is important in proofreading. It helps in proofreading, okay? Newly synthesized DNA. That means 3' to 5' activity help in the proofreading of newly synthesized DNA. I will show that in figure 1. I will show you that the figure in my next slide. If DNA poly 1 has just added the wrong nucleotide to a growing DNA chain, this nucleotide will not base pair properly with its partner in the parental strand and should be removed. We know the base pairing rule, that is the G always bind with C and A always bind with T with help of double bond. According to Pol1, pauses the 3' to 5' exonuclease removes the mispaired nucleotide, allowing replication to continue. This greatly increases the fidelity or accuracy of DNA synthesis. And that's the figure. The first one shows the pink structure, that is the adenine. Here try to bind with the G, that is the guanine, but it is the mispairing. So Pol1 stop the synthesis and removes the mispaired base, that is the adenine, and repair it or proofread it. Then the process of synthesis is going on. Here's the descriptive part of the figure. You guys can read it. So the 5' to 3' exonuclease activity allow Pol2 to degrade a strand ahead of the advancing polymerase. So it can remove and replace a strand all in one pass of the polymerase, at least in vitro. This DNA degradation function is useful because Pol1 seems to be involved primarily in DNA repair. Now we know that DNA Pol1 helps in only DNA repair, but at that time the whole thing is assumption, including a removal and replacement of RNA primers, for which destruction of damaged or mispaired DNA or RNA primers and its replacement by a good DNA is required. In figure 2, I show the structure. Illustrate the process for primer removal and replacement. That's the figure 2. That's the descriptive part. That is the removing primers and rejoining nascent DNA fragments. That is, the nascent means the newly synthesized. in the figure 2, that is the part A, there are two adjacent progeny DNA fragments, the right hand one containing an RNA primer, that is the red in color, the RNA primer in red in color, at the end of 5' end. That in this strand, the progeny strand, this is the 3' end, so that will be the 5', that will be the 5'. The two fragments are separated by a single stranded break called NEET. In DNA fragment, the break is known as a nick. In the figure of B, DNA polymerase 1 bind to the double-stranded DNA at the nick. So here, the bubble-like structure, it shows the just assume that that is a DNA poly 1. It bind the nick portion. The 3' to 5' exonuclease and polymerase activity of DNA polymerase 1 simultaneously remove the pair and fill the resulting gap by extending the left hand DNA fragment rightward, just like parent, left right. So here the fragment of the DNA goes in the rightward direction from left. In the D part, DNA ligase, not that here in the C part, the C part is that the polymerase structure moved in the right direction. In part D, DNA ligase seals the remaining nick by forming a phosphodiester bond between the left hand and right hand progeny DNA fragment. I told you guys in the first part of DNA replication, prokaryotic DNA replication, that DNA ligase always help to joining two break strands. Another important feature of POL1 is that it can be cleaved by mild proteolytic treatment into two polypeptides. A large fragment that is known as Klenow fragment. The Klenow fragment is also abbreviated as KF. K stands for Klenow and the F stands for fragment, which has the polymerase and proofreading activities in the 3' to 5' direction, and a small fragment with the 5' to 3' exonuclease activity. The Klenow fragment is frequently used in molecular biology when DNA synthesis is required and destruction of one of the parental DNA strand or in the primer is undesirable. Now take an example. The Klenow fragment is often used to perform DNA end filling and can also be used as the sequence of DNA. On other hand, the whole POL1 is used to perform a NEET translation to label a probe in vitro, because NEET translation depends on 5' to 3' degradation. So it shows the 5' to 3' degradation and the 3' to 5' degradation. So we can tell that DNA POL shows the exonuclease activity in the 3' to 5' or 5' to 3' direction of that's it. It shows obviously it will work in the moving fork of prokaryotic DNA. Now we will discuss about the structure of DNA polymerase 1. Just wait a minute. Thomas Days and his colleagues determined the crystal structure of Klenow fragment in 1987, giving us the first look about the fine structure of a DNA synthesizing machine. The most obvious feature of the structure is a great cleft between two A helices. It's not A helix, it will be the alpha helix. Just correct it. That will be the alpha helix. This is the presumed binding site for the DNA that is being replicated. In fact, all of these known polymerase structure, including the structure of T7 RNA polymerase, are very similar and have been linked to a hand. So our hand contains fingers, thumb, and a palm. So we can guess that DNA poly, the structure of DNA poly also have those domain. In Klenow fragment, one alpha helix is a part of finger domain. So alpha helix contains the part of finger domain, the other part of the thumb domain, and the beta plated sheet between them is a part of palm domain. So beta plated sheet, and the remaining one is thumb domain. The palm domain contains three conserved aspartate residues that are essential for catalysis. They are though coordinate magnesium ions that catalyze the polymerase reaction. So here a question arises, that is the cleft in the polymerase structure really the DNA binding site? Just wait a minute, I have to erase the, okay, here's the question. So the answer is to find out that stage and is equally strand to another DNA polymerase, the tag polymerase. So what is tag polymerase? The tag polymerase obtained from the thermophilic bacterium, that is Thermus aquaticus. They made a co-crystal of tag polymerase and a model double-stranded DNA template containing a 8BP and a blunt end at the 3' end of the non-template, that is the primer strand. Its polymerase domain is very similar to that of Klenow fragment, so much so that it is called the KF portion. Now Klenow fragment portion of the enzyme. In figure 3 shows the results of X-ray crystallography studies on the tag polymerase DNA complex. The primer strand, that is the red one, that's it, the red one, the red strand, the primer strand has its 3' end close to the 3 essential aspartate residue in the palm domain. The red ball-like structures, here the red ball-like structure, those are the, I think you guys can see it, the red ball-like structure. That's the aspartate residues in the palm domain, but not quite close enough for the magnesium ions to breach between the carboxyl group of the aspartate and the 3' hydroxyl group of the primer strand. Thus, this structure is not exactly like a catalytic productive one, perhaps in part because magnesium ions are missing. So, in figure 3, we see that there is one O helix and the another one the I helix. The O helix and the I helix of the finger and thumb. So it is the finger domain, and that is the thumb domain. So the remaining one is palm domain. The red one, the red one is the template strand, and the orange one is non-template strand. That's it. The 3 essential aspartate side chains in the palm domain are represented by small red balls near the 3' end of the primer strand. So there comes the another question, that how scientists confirm that there are other DNA polymers except that the DNA poly 1. In 1969, Paula DeLucia and John Cranes isolated a mutant with a defected in the pol A gene, which is encodes pol 1. So DNA pol A encode DNA pol 1. So if a mutant does not contain the pol A gene, so it does not have DNA pol 1. DNA pol 1 is absent in that mutant. This mutant pol A1 lacked pol 1 activity, yet it was viable, strongly suggesting that pol 1 was not really the DNA replicating enzyme. Pol 1 is not DNA replicating enzyme. Instead, pol 1 seems to play a dominant role in the repair of DNA damage. It fills the gaps between the damaged DNA is removed. When the damaged DNA is removed, the finding that pol 1 is not essential sparked a renewed search for the real DNA replication or replicase. And in 1971, Thomas Kornberg and Malcolm Gifter discovered 2 new pol activities, that is the DNA pol 2 and DNA pol 3. So that's it for today's video. We studied the structure and function of DNA pol 1. In our next video, we will discuss about DNA pol 2 and DNA pol 3. So thank you. Don't forget to hit the like button below the video. Please subscribe to stay tuned on mushrooming your knowledge.